Regarding viral genetics, the first genetic the first genetic type of virus is recombination, the process which virus use in order to increase its virulence or uh, in order to transfer its uh, DNA into the respective progeny. So recombination means there is an exchange, and exchange of what? Exchange of genes, exchange of nucleic acid, exchange of chromosome, exchange of DNA. And recombination, as we have known that the recombination always happen between two homologous chromosome, and this recombination is very much in the viruses which are non-segmental, and these are double-stranded. Obviously we need homologous chromosome, two homologous chromosome. So non-segmented double-stranded DNA viruses via crossing over. So all the double-stranded DNA viruses which are non-segmental, they exchange their gene via crossing over, and in this way they can do recombination. And after recombination we have the reassortment. Re means there is also an exchange, and in this exchange there is assortment. There is exchange of the whole segments, and these segments are obviously in the segmental type of viruses. And because of this segmental exchange there is a antigenic shift. S for segment, one S for segmental and one S for shifting, antigenic shift, which lead to pandemic, like the avian influenza and the human influenza, and they recombine to make a coronavirus, which is a type of SARS COVID-19. The next one is complementation. Complementation means whenever there is a virus, for example vitamin D, vitamin D, for example hepatitis D virus, and this hepatitis D virus has nothing to do when it is alone in the environment, but it gets its virulence whenever it get co-infected with hepatitis B. For example, hepatitis D, it infect only when it gets complement, and in complement it gets a functional protein, and this functional protein is actually the surface antigen which coats it. And now hepatitis D virus is encoded by a complement gift of functional surface protein from the hepatitis B virus. So now hepatitis D can cause double infection, like it can cause the complement, the surface antigen complement, it serves both the hepatitis B and hepatitis C. Means it not only help to cause infection in the patient by hepatitis D virus, but also it causes hepatitis B. So that's why we always said that B is bad, because B can cause D co-infection, so it serves both. So this is example of complementation. This is how viruses genetics happen. And then we have the last type is phenotypic mixing. And this phenotypic mixing is some kind of mixing mixing of a phenotype. And the mixing is happen when one cell is infected by two different viruses, and this is simultaneous infection. Nowadays, during this pandemic, I have seen a lot of information on the internet that a lot of patients are going to die by two different strains of coronavirus, but approximately the delta strain or the beta strain of women is infected by that kind of. So that particular event is an example of phenotype mixing, in which there is two different strains of coronavirus. It infect a same person cell, and in this way it can acquire the progeny, the first progeny, which produced by these two strains. It has the same genome of one strain, for example virus A strain, but it has a surface protein coat or surface antigen, surface protein from the virus P. So now this progeny is very much fatal, very much lethal to cause infection in that person. So this surface, or we have known that the surface proteins always, the surface protein coats always cause infectivity. So the infectivity or the tropism always follows the particular type of virus which gives or which donate that surface protein. So in this progeny, in the variant of the coronavirus, in that female which is suffered from two different strains of coronavirus, so the infectivity is actually because of the strain which gives the code, but the genetic code remains the same of a particular variant. So in the progeny, the genome is of a virus A, and the surface protein is from different virus. But when that person's having the virus progeny, it again replicates. So the progeny which are produced subsequently, or the next progenies, they have revert or remains the same to their standard or to their parental genome. What does it mean in phenotypic mixing? Whenever there is two strains infect a single cell, then in the first progeny, one cell, two strains come across and they produce a progeny. They mix their genetic material and produce a progeny. So that progeny has a genome of one variant and has a surface proteins of another variant, and it can causes a lethal effect or life-threatening effect in the particular person. So after causing these effects, now the virus again replicate and produce another progeny. So in the next generation, and in the next progeny, the virus, the progeny having the genome from its parental type of genome. For example, the progeny has the parental genome of virus A. So this type of genetics is known as the phenotypic mixing, and it have been seen in these type of situation. For example, the corona situation. So this is the four types of viral genetics. After this, we have bacterial genetics. In the bacterial genetics, the first type is transformation. And what does transformation mean? Transformation means there is a transform of one thing into another. So transformation is actually the process in which there is a transfer, and expression of what? Of newly transferred exogenous naked environmental newly transferred bacterial DNA. So this is bacterial genetics, and exogenous naked bacterial DNA, which is picked up by a competent bacteria. For example, B. shine bacterias, Bacillus, Streptoneumoniae, Haemophilus influenza, and Neisseria. They are the pretty much competent bacteria. They take up bacterial DNA from the environment for the naked DNA, and they take up that DNA. They transfer it into their own genome. They express it, and this process is known as transformation, and make them acquire virulence from these bacteria. So these B. shine bacteria, they make non-virulent virus to be a virulent strains or virulent virus, and this transformation is inhibited by or is prevented by a nucleases, which are deoxy. Obviously we are talking about the deoxyribonucleic acid, so we need deoxyribonucleases which dissolve these naked DNA. So this is transformation. The next process is conjugation. Conjugation means we conjugate something, and in conjugation we have two things: F positive into F negative, or HFR into F negative. Conjugation is the process in which there is a bridging mate in which there is F positive, F negative mating. This is mating, bridge mating. F positive means fertility factor. F positive means fertility factor. And if a bacteria has this fertility factor, it means that it is fertility factor positive. And if it doesn't have any fertility factor, then it is fertility factor negative. What is this fertility factor? It is actually the fertility factor is actually the code, the genes present in the plasmid, which makes them fertility factor is actually the single stranded plasmid genes, which causes sex, pili, and transformation or mating process, conjugation happen. So if one cell is fertility factor positive, how this cell make a fertility negative bacteria into a fertility positive cell? By the by a bridged mating in both. So now the fertility factor positive single stranded plasmid gene is transferred to the fertility negative bacteria, and in this way both acquired the genes for sex, pili, or conjugation. So this is pretty much a simple process, but complexity happens when a bacteria which first a bacterial DNA of a fertility positive cell, a fertility positive bacterial DNA first incorporate, uh, for example, a fertility positive fertility positive plasmid DNA is incorporated into the bacterial DNA. We have known that there are two types of DNA. First, we have the plasmid DNA, and the second one is the nuclear DNA or the bacterial DNA. So, if we directly transmit the plasmid gene into one cell into another without using the nuclear DNA, then it is known as conjugation and fertility positive into negative, making both of them acquired that conjugation process. But if we do not use a direct process, rather we bypass a process, we just transmit that fertility positive DNA of single stranded DNA of the plasmid and incorporate, important word, incorporates the fertility positive plasmid DNA into the bacterial DNA. Now this whole cell has become a recombinant variant, and this recombinant variant is known as high frequency recombinant bacterial DNA or bacterial cell. So now this cell is now this bacterial DNA is now transferred to another cell, which is incorporates. Now it transfer into the cell which is fertility factor negative or fertility factor deficient. So that fertility so now we transfer this high frequency recombinant DNA into the F negative cell to make it conjugate, but the cell remains F negative. In simple words, high frequency recombinant means that the plasmid fertility factor genes are now incorporate into the its own bacterial DNA, which is fertility positive. Now this bacterial is using the bridge mate, and this bridge mate is now transfer not only the fertility factor single stranded genes, but also transfer some flanking sites of the bacterial genes. So after transferring bridge mating to another F negative cell, after transferring single stranded F positive genes along with bacterial genes, now that become a recombinant cell. Means it has both genes of F negative and F positive. So it becomes reinactive, but it remains F negative. So this process of conjugation is known as high frequency recombinant to F negative transformation. The next one is we have transduction. We have two types of transduction. First one is generalized, which is actually a packaging error, and second one is specialized, which is a axonal defect. In generalized transduction, we have first there is an infection of a lytic phage. In specialized, we have infection of lysogenic phage. Two different phages. Lytic phage infect the bacteria and cleave its DNA, cleave bacterial DNA. But in lysogenic phage, it doesn't. Yes, it infects the bacteria, but it doesn't cleave the bacterial DNA. Rather, it integrate into the bacterial DNA. In the lytic phage, in the generalized transduction, it cleaves the bacteria, then it order packaging. So now there is a defective packaging. The new progeny, defective packaging. The new progeny is not only package the viral DNA but also packages the bacterial DNA, and that bacterial packaging, defective bacterial DNA packaging into the new progeny So now this progeny is a recombinant type of progeny. So it infect another bacteria and releases its DNA. So that DNA goes into that bacteria and incorporate and express, and now they acquire virulence or antibiotic resistance. This is the process of acquiring virulence or antibiotic resistance. In contrast to this, we have no cleavage of bacterial DNA. Rather, we directly integrate the lysogenic phage into the bacterial DNA, and now the bacteria is going to excise. So there is a defective excision. The phage is going to excise. There is defective excision. So the excision not only excise the phage or viral DNA, but also it excises the flanking sites of the genes of bacterial DNA, and now the progeny is get recombinant and it infect others and make them acquire virulence. And we have 4 A, B, D, C, Ds. We have 5 types of different bacteria which acquire lysogenic phage transduction, in which we have the group A Streps, which has the erythrogenic toxin, which acquire virulence by this acquired virulent toxins, erythrogenic toxin. We have the diphtheria toxin, Shigella toxin, cholera toxin, and botulinum toxin. So this is a particular type of bacterial genetics. So this is all about transduction. The last one is transposition. The last one is transposition. Transposition is what? Transposition is actually specialized transposition, the process of transpose, the process of jumping of what? Jumping of transposon, jumping of transposon. Transposon is actually a specialized segment of DNA segment. which has, for example, we say a particular type of virulence. For example, in the enterococci, we have a segment known as bank A, and it has a particular type of uh, genes which create plasmids with multiple drug resistance, MDRs, multiple drug resistance. So enterococci bacteria use this DNA in order to in order to generate multiple drug resistance. So this transposon segment is copy itself, it excise itself, it integrate itself. It copy itself, it excise and integrate itself, and it incorporates within enterococci species or enterococci its own plasmids and chromosomes or its own DNA. Not only it do this for itself, but also it does this for staph aureus and make them also a multiple drug resistant strains. So in this way, because of this transposition, the new MRSAs or vancomycin resistant strains are going to evolve because of this transposition MDR resistance. So this is all the bacterial and viral genetics.